Welcome to LankanTutor's Biology 2.11, and we're going to be doing animal tissues. Last time in 2.9 and 2.10 we were talking about plant tissues and the differences between them, whether it's a simple plant tissue or a complex, also known as a permanent plant tissue, the xylem, the phloem, and everything. So it's time to talk about the animal tissues. We already know what a tissue is. A tissue is a group of physically linked cells which have a common origin and which are designed for a specific function. Animal tissues can be divided into four parts. Well, four kinds of tissues, and you can divide them into three types depending on their origin. Now, first of all, we're going to divide it into epithelial tissues, and then we have connective tissues, and then nervous tissues and muscular tissues. These tissues have different origin, specifically ectodermal, endodermal, and mesodermal. What you need to know is epithelial tissues can be ectodermal, endodermal, or mesodermal in origin. Connective tissues are only mesodermal. Nervous tissues are only ectodermal, and muscular tissues are only mesodermal. In 2.11, what we're actually going to be talking about is epithelial tissues. There are a few kinds of epithelial tissues. Some are simple and some are complex, and we'll give a brief description of each one of those and how to draw them and their functions. So the epithelial tissue is a single or multi-layered sheet. It's lined in the internal or external free surfaces of the body of an organism, and the main feature of the epithelium is that it has a basal membrane in which the cells can rest on. There are small amounts of cement or cementing material which keeps the cells together, and there are no blood cells directly in the tissue, but there is a supply of blood to the basal membrane. So you'll get a better idea of it once we start drawing the structures. Now, before we go into drawing, let's just divide the epithelial tissues. It gets divided into simple epithelium and compound epithelium. The only difference between the simple and the compound is that the simple only has one layer of epithelium, whereas the compound has more than one layer. The simple epithelium can be divided into squamous epithelium, cuboidal epithelium, columnar epithelium, and pseudo-stratified epithelium. And the compound epithelium is stratified and transitional. You're going to wonder why the pseudo-stratified is a simple epithelium and why the stratified is a compound epithelium. Don't worry about that. We'll get into it during the description. I'm going to go ahead and start drawing now is the simple squamous epithelium. The way you can find out if you see a drawing, it is generally a thin and very short layer of epithelium. The main use of the simple squamous epithelium is to facilitate diffusion. So things have to pass through that. That's how you remember simple squamous. And you can get the idea when you hear the word squamous of it's a really thin and flattened epithelium. So it has all the basic structures. It has a basal membrane, and remember this word, it's a tessellated structure. It's called tessellated because the structure is not regular, it's irregular. There are different heights in different areas, and it's like this giant mesh. The nucleus can also be seen in the epithelium. However, the nucleus is also a bit flattened to fit inside the cell. And again, the main function of the simple squamous epithelium is to facilitate diffusion, and it is also the lining of various organs. Now, here are the locations that you actually need to remember where you can find the squamous epithelium. It can be found in blood capillary walls, where blood cells need to move from one place to another. It can be found in the lining of the alveoli sacs in lungs, because that's another place diffusion needs to take place. The heart chambers are lined with simple squamous epithelium, and also the Bowman's capsule, which is found in the nephron, where ultrafiltration takes place, is also lined with simple squamous epithelium. Next, we get on to the cuboidal epithelium, and as the name implies, it looks like a bunch of cubes. It's generally found in glands. So the cuboidal epithelium also has a central spherical nucleus, which is rather large, and a basal membrane like every single epithelium. From the surface, you can either see them as a pentagon or a hexagon in shape, which means it has either 5 or 6 sides. Now again, you need to remember where the locations of each of the epithelium are. The questions are generally going to come like, what is the type of epithelium you can find in this location? So for example, if they say thyroid gland, the answer would be a cuboidal epithelium. The thyroid gland, salivary gland, and sweat glands, and basically anything which has a secretory function, the simple cuboidal epithelium could be found. Also, things like the lining of the pancreatic duct and parts of the nephron and the salivary gland is covered by simple cuboidal epithelium. Now remember, when they say parts of the nephron, this does not include the Bowman's capsule, which is lined by squamous epithelium. And the third type of epithelium we're going to be talking about is the simple columnar epithelium, and again, as the name implies, it is just like a column. This is one of the most common kind of epitheliums that you can see in the human body, and also remember that it's a bit different from the cuboidal because there's a bit more to learn. Now, you can see here in the structure that they are really tall compared to all the other cells. It's a bit weird because the nucleuses are near the basal membrane. So when you're drawing the structure and you're drawing a little circle to indicate the nucleus, make sure you draw it as low as possible, but don't get it in contact with the basal membrane. Just leave it slightly above it. And they have several mucus secreting cells or goblet cells, and there's this massive invagination in it, and you can see that clearly. And there are also cilia you can find on the cells. Cilia is basically like hairs-like structures, and you can also see microvilli in some forms of cuboidal epithelium. Remember that they are slightly narrower than the cuboidal cells, but they are extremely longer. The function of the microvilli inside the epithelium is to increase the surface area, to increase the rate of reactions. And again, we're going to be moving on to the locations. The locations are, the first one is the human alimentary canal. That is the canal that sends food from your mouth to, well, your rectum. You can find columnar epithelium with secreting cells and brush borders, or should I say microvilli, all over the alimentary canal. You can see it in the lining of the stomach. You can see it in the fallopian tubes. You can see it in the central canal of the spinal cord. And you can see it in the brain cavities, also known as ventricles. Now, don't get these ventricles confused with the ventricles of the heart. And then we go to the pseudo-stratified epithelium. Now I'm going to start drawing it and explain a bit about it. At first glance, you're going to realize that there is more than one layer of cells. And you're going to assume that, isn't that a compound or complex kind of epithelium? Because it has more than one layer of cells. But if you look closely, you can actually see that, again, as the term implies, pseudo, meaning it's not the actual thing, that the first layer isn't actually a layer. There is a germinal layer, and it looks a bit like a actual stratified epithelium, but the cells aren't equally aligned, and it looks like there are two different layers of cells, but it's just one. And all the nucleuses are not in the same level. Some of them are in the same level and some of them aren't. It's completely random. So that is the pseudo-stratified epithelium. Now we're going to talk about the actual stratified or the compound stratified epithelium. Now these tissues are made up of several layers of cells, which means it's thicker than simple epithelium. They're relatively tough and they are impervious, which means that they don't allow things to go through, unlike the exact opposite, which would be the squamous epithelium. Now, because it has a lot of cells, a lot of layers of cells, the basal layer is actually called the germinal epithelium, the lowest layer, and they are capable of division, and that's why they're termed the germinal epithelium. There is a basement membrane. It's just like everything else, except that remember that the direction of growth is upwards, so the newest cells would be in the germinal epithelium, and on the very top there are keratinized stratified epithelium, and then there are non-keratinized ones as well. Don't worry about that. Just remember that some of the stratified epithelium, they have keratin deposited on top. As more layers grow, the germinal epithelium gets pushed up and a new germinal epithelium forms. Now, you need to remember that the stratified epithelium are divided depending on the type of the cells inside it. So that means there are stratified squamous, that means there are stratified columnar, and that means there are stratified cuboidal. The one we just drew now was a stratified cuboidal epithelium, but you can imagine how it would change as it becomes columnar and squamous. So the stratified squamous epithelium could be found in the human skin, the epidermis, and in these cells, the top cells contain a protein layer called keratin. So the human skin has keratin on it. Don't mix keratin with chitin. There are two different kinds of proteins. If you don't remember the proteins, there is a unit on that we covered in the first part. I think it was 2.2. And then there are non-keratinized stratified squamous epithelium. It might get a little tricky. So we divided epithelium into compound and simple, and then we take the compound and we divide it into a transitional and stratified, and we take the stratified and we divide it again into stratified squamous, stratified columnar, stratified cuboidal, and then we take the stratified squamous and we divide it again into a keratinized and non-keratinized way. So the keratinized stratified squamous epithelium would be, the example would be the human skin, and for the non-keratinized version it would be in the esophagus, the buccal cavity, and the female vagina. And then there's the stratified columnar epithelium, which are generally located in the mammary glands, and the stratified cuboidal epithelium, which are found in some sweat gland ducts. And then we move on to the transitional epithelium. Now, you don't really need to learn a lot about transitional epithelium because it is one of the most complex types of epithelium. It consists of 3 or 4 layers of cells, and the top layers are not peeled off. This is because the main function of transitional epithelium is elasticity. It has the ability to stretch, like the human urinary bladder and the renal pelvis is made out of transitional epithelium because they can expand. Again, epithelium can be divided according to different things, whether they are simple or compound, or whether they're keratinized or not. They can also be divided by function. Some of them are compound, some of them are glandular, and some of them have exocrine functions, and some of them have endocrine functions. Anyway, that is all you really need to know about epithelium. But there is one little bit that people teach during this part about glands, about the shapes of glands, and well, the shape of the duct, but that really isn't necessary. If you want to know, I'm just going to go ahead and talk about that in the next minute, but remember, it is not, you'll probably never have to learn this, or you probably won't get any questions based off of this. So I'm just going to go ahead and draw these structures, the types of glands, or the types of ducts. Just remember that there's simple tubular, there's simple coiled, there's simple branch tubular, and then there's compound tubular, and then there's simple alveoli, and then there's simple branched alveoli, and then there's compound alveoli. Just remember it if you think you need to, but otherwise it's not that necessary. So we started animal tissues and we completed the first part, which is epithelial tissues, and next time in 2.12 we will be going into connective tissues. Now, connective tissues is a massive part because it contains everything from bones to blood to the loose connective tissues, and there is a huge huge part. We might not be able to complete it in one unit or in one video, but stay tuned. We will do it as fast as we can. So thank you guys for watching. We will see you again next time. And if this video did help you out, don't forget to share it with one of your friends. And if you're not watching this on LankanTutor.com, we suggest you do so, as there are a bunch of written tutorials for you guys that would help you study further. So until next time, have a great day.